The way we've been able to characterize cells in culture, cells in the lab, living cells that we rely upon to do basic research, to do translational research, has really grown just amazingly in the last 20 years. If you were to jump on a time machine 20 years ago and come to 2023 and see the way we can characterize cells, the way we can identify them, it would almost look like science fiction to you today. We've gone from characterizations of cells which would look at bulk populations of cells that we would homogenize. So all the cells would look the same because the methods weren't granular enough. We've gone from that kind of scenario down to the scenario where we can look at a single cell. We call these single cell technologies, single cell sequencing technologies that have grown in ways that are just amazing to us. In the last couple of years, we've added granularity to that. We can characterize that single cell, but we can add the component of spatial recognition. We can say this cell that we now know, we can identify, we can say this is what this cell is doing, what it is. We can say that cell came from here. We know exactly the place in the tissue and the broader system that the cell came from. What we wanted to do with our method, which we call segmentation by exogenous perfusion, or SEEP, is add another bit of detail to that granularity, which is the added component of not just spatial recognition, but the ability to understand how that single cell had access to its broader environment. And that's really important. Yeah, that really defines what a cell is and how it's going to respond to changes in its little microenvironment. And we did this using what is a relatively straightforward technique. We added the component of a cell-penetrant dye. So just think of any kind of fluorescent dye that anybody in a lab would use. And we added that at the external domain, the exogenous space of a three-dimensional model. And that dye, over time, gradually perfuses or seeps through those three-dimensional models. When we define the rate at which it will perfuse or seep through those models, we can go back in later and then say this cell had this much fluorescence in it, this much dye in it. So it must have come from here. And then when we do the identification methods, in this case transcriptomics or RNA-seq, we can say this cell with this specific identity came from this location and had this much access to its exogenous environment. So that was the real goal of the work that we performed that's being revealed in this paper.